Okay, so welcome to our new video presentation for the continuation of the lesson 2 in Research 2. And for this topic, we will be dealing with the gathering or the methods on how we gather data in quantitative research. Now, in quantitative research, we all know that quantitative research uses numbers. Okay, it uses quantities as its main data. So meaning to say, if quantitative research concerns with the gathering of data in a quantitative manner, or let's say in a numerical form, meaning to say, the data in quantitative research requires quantitative analysis. Quantitative analysis. Okay? So what is quantitative analysis? So since quantitative research concerns with the numerical data, it is subjected to quantitative analysis. So quantitative analysis, meaning to say, it is the analysis involving numerical data. Analysis involving numerical data. And this numerical data, which is analyzed and interpreted, are actually derived from the different statistical treatments. Okay? But how do we exactly gather data in quantitative research? Where do we get these numbers to be subjected for quantitative analysis? Okay, so let us enumerate, okay, the four methods in gathering data of data in quantitative research. So it actually uses four methods. And these four methods can be added, or let's say, it can be varied from time to time. Okay? So it uses the following methods. Number one, it uses sampling methods. Sampling methods. Okay. Then number two, it also uses experimentation or experiments. Number three, it also gathers data through the use of surveys and questionnaires. And lastly, it gathers data using inferential analysis. Inferential analysis. Okay? So these four methods are the actual four main methods in which where we can gather data when we are conducting quantitative research, and we will discuss them one by one. Okay? So let us first start with the sampling methods. Okay? So sampling methods. What do we mean by sampling methods? Actually, sampling methods are very unique in quantitative research because by the use of sampling methods, we can identify one characteristic of quantitative research, and that is by using samples derived from population. So meaning to say, sampling methods is a method whereas it pertains to the deriving of samples from a given population. Okay? And this derivation of samples from a given population is actually used in the process of inferential statistics. Okay? It is used in inferential statistics. Okay? So that is the main definition of using the sample methods. Now, let us have a deeper understanding or deeper definition of the sampling methods. So sampling methods. Okay, sampling methods. So we said earlier that in sampling methods, we derive samples from population. Okay? We derive samples. Now, samples is represented by the small letter n. Okay? And this sample is derived from population. Population is symbolized by the big letter N. Okay, so what do we mean by samples and what do we mean by population? Now, the samples, okay, from the name itself, samples, so meaning to say, these are the chosen subjects. Okay, the samples are the chosen subjects. So meaning to say, the subject of the experiment. While the population is the overall count, where the sample belongs. Okay, where the sample belongs. So, the samples, these are the chosen individuals or the chosen respondents, the chosen experimental units out of the number of the population. Now, meaning to say, if the samples are the chosen subjects derived from the population, the samples will represent the population. Okay, the samples will represent the population in an experiment. Okay, so, however, when we use sampling methods, it is very inevitable for us. Okay, it is very impossible to do not encounter weaknesses or to do not encounter such type of disadvantages. So, the disadvantage in using sampling methods is that, okay, the advantage in using sampling methods is that the effects prevalent in the research only apply to the samples rather than the whole population. So meaning to say, the weakness, okay, sometimes the effects of the, okay, the effects of the prevalent research, sorry, the effects of the prevalent research okay, only apply to the present sample rather than the whole population. So meaning to say, for example, we conduct a research. So let us have this illustration to understand this disadvantage. So let's say we conduct a research. Okay, we conducted a research. And this research used different samples. Now, these samples are, let's say, subjected to the experimental setups. Now, since these samples represent its whole population, okay, meaning to say, the experimental setups can apply to the samples with the ambiguity, or let's say with the doubt that these experimental setups can also apply to the population. What do we mean by that? Meaning to say, the experimental setups, there would be the doubt that it will only apply to the samples rather than the population, because the main essence of using sampling methods is to reduce the cost and to reduce the amount of the experimental units. However, the disadvantage is that sometimes the effects of the experimental setups applied on the samples only apply to these samples rather than the whole population. Okay? So, that is the main disadvantage. Okay? So, let's say we have a species of bacteria. Let's say we have Pseudomonas aeruginosa. And this Pseudomonas aeruginosa bacteria actually has different, or let's say has a large population. So, we cannot test our experimental setups to all population of Pseudomonas aeruginosa. So, we just obtained samples of this bacteria. Now, when we apply the experimental setup to this sample, it produced results. However, in the next trial of the results became unreliable. It became inconsistent. So that is what we mean by this disadvantage. Okay, the results only apply to the samples. But when the result is applied to the population, it will produce unreliable results. Okay? So that is why research or quantitative research is very repetitive in nature. It tries to validate the reliability of the results. Okay? So that is the main essence of sampling methods. Now, since we have, let's say, described sampling methods, okay, how do we actually do, or how do we actually, let's say, execute sampling methods? So let us have this example. Okay? So in continuing, let us have this example. We are still on the method number 1, sampling methods. Okay? So let us have a situation in which we will apply sampling methods. Let's say in a population, okay, we have a population of 1,000 bacterial cells. Let's say we have 1,000 bacterial cells. Okay. We only pick out 3 sample species. 3 sample species. Okay. And these sample species are actually the following. We have Escherichia coli. We have Staphylococcus aureus. And we have Pseudomonas aeruginosa. So these are the 3 bacteria. Okay? Or the 3 bacteria that are sample. Okay? The sample from the 1,000 bacterial cells. Now, each sample species, okay, actually have their own population. So let's say we label this Escherichia coli as N1, Staphylococcus aureus as N2, and Pseudomonas aeruginosa as N3. Now, the population of Escherichia coli, okay, resulted to 500. Then the population of Staphylococcus aureus resulted to 600. Then the population of Pseudomonas aeruginosa resulted to 800. Okay? However, in your let's say research, in your quantitative research, you only have a research design whereas you only use 6 setups. Okay? So in your research design, you decided to use 6 setups only. And these 6 setups contain 3 trials per setup. Okay? 6 setups, then 3 trials per setup. Okay? So meaning to say, we have 6 setups and each setup has 3 trials or setups. So meaning to say 6 times 3 is how much? We have 18. Okay? We have 18. However, this 18 only applies to one, or let's say 1 population. Okay, 1 population only. So meaning to say, how many population do we have? We have 3 populations. So 18 times 3. Okay? 18 times 3, we have 54. So, meaning to say, combining all this population, all these used bacteria, we will have 54 samples all in all. Okay, 54 samples all in all. Now, how many samples do we need for each bacteria? So, we have sample number 1, sample number 2, and sample number 3. Okay? So, sample 1 pertains to the Escherichia coli, sample 2 pertains to Staphylococcus aureus, and sample 3 pertains to Pseudomonas aeruginosa. Now, if we have 54 samples all in all, how many samples will be derived from each bacterial population? Okay? So, since we have 54 samples and we have 6 setups with 3 trials each, meaning to say we will have 18 samples for each bacteria. Okay? So, I hope you get that. Okay, so let us summarize this one. In a 1,000 bacterial cell population, we only obtain 3 sample species. And these 3 sample species is Escherichia coli, Staphylococcus aureus, and Pseudomonas aeruginosa. Now, each sample species has its own population. Okay? Escherichia coli has a population of 500. Staphylococcus aureus has a population of 600. And Pseudomonas aeruginosa has a population of 800. Now, in your research design, you only have 6 setups. And each setup is composed of 3 trials. So, meaning to say, 6 times 3, we have 18 setups all in all. Okay? And these 18 setups will be multiplied to 3. Why? Because we have 3 different bacterial sample species. But, this overall count of sample will apply to all of these bacterial species. So, meaning to say, we still need to divide the different samples. So, Escherichia coli will be further divided into 18 samples, while Staphylococcus aureus will be further divided into 18 samples as well, and Pseudomonas aeruginosa will have 18 samples as well. Okay? And these 18 samples will comprise to the sample count of 54 out of 1,000 bacterial cells and out of 3 sample species. Okay? So that is the main essence of sampling methods. We try to derive different samples from a population. Okay? Same as here, we derive different samples out of the different populations. And these samples will be used in our experiment in the 6 setups. Okay? So that would be the main essence or the main, let's say, the main idea of using sampling methods. Okay? Now, in continuation with the 4 methods of gathering data in quantitative research, we have number 2. We have experimentations. Okay? So, we have experimentations. So, in experimentations, okay, linking sampling methods with experimentations. How do we define experimentations? So, experimentations are actually, okay, the process in which we form treatments. Okay, forming treatments. From what? From the collected samples. From the collected samples. So, in our example here, we use sampling methods in which we obtain these following samples. Now, these following samples is actually subjected to these 6 setups with 3 trials per setup. Now, these 6 setups with 3 trials per setup actually refer to the treatments in your research design. These are the treatments, and these treatments will serve as your basis for the experiment. Okay? So that would be the main essence of experimentation. Also, in experimentation, since we have these different setups, we have these varied setups, meaning to say, in experimentation, it involves manipulation of variables. Okay? Since we have these 6 setups, it vary from each other. It vary from one another. That is why it is called treatments. It has variation. And this variation actually pertain to the manipulation of variables. Okay? And we do that under the gathering of data or the method of gathering data of experimentations. Okay? Also, since we involve the manipulation of variables and we involve different treatments, it is also safe to say, or it is also safe to define that in experimentation, it makes use of analytical observation. Analytical observation. Okay? So that is the main essence of experimentation as a main method of gathering data. It involves manipulation of variables, it involves analytical observation, and it involves the use of different treatments. The different treatments from the collected samples. Okay? So that is experimentations. Now, what do we actually mean by treatments? So let us have this example. Okay? So to discuss experimentation even more, let us have a definite, or let us have a concrete understanding of the treatments. So in making treatments for experimentation, let us have this treatments. So let's say we use the variable Escherichia coli. So this variable is the bacteria. It is the experimental unit. Okay? So this bacteria has a population of 500, as we mentioned earlier. And from this population of 500, we gathered samples, and that sample is 18 samples. Now, to get the treatments, we will have to compute for the sample trials. And what is the formula? Sample trials is equal to the number of samples over the number of trials. Okay? And we have 18 samples, and let's say we would want to have 3 trials for each sample. So we will have 6 setups. Now, since we have 6 setups already, okay, let us try to plot that setups. Okay, we have 6 setups. So let us name the setups. A, B, C, D, E, and F. We have 1, 2, 3, 4, 5, 6. And each setup will have 3 trials. So we will have trials, then trials 1, 2, and 3. Okay? Now, since experimentation deal with the manipulation of variables, especially the independent variable, each setup will be defined by the independent variable. So let's have first setup A. Let's say setup A will have 25% independent variable. Setup B will have 50% independent variable. Setup C will have 75% independent variable. Setup D will have 100% independent variable. Setup E will have the commercial variable or the commercial brand. What is the commercial brand once more? It is the positive controlled setup. Okay? Then the setup F, okay, setup F, it will have the negative controlled setup. Okay, the negative controlled setup. Now, so we have these following setups already. We have defined it. Let us form the treatments. So let's say, after you have experimented on this dependent variable, okay, in the Escherichia coli of that, let's say, experimental unit, you derive with the following results. We have 30.0, 29.1, we have 31.0, and the average would be 30.03. Let's say we have 35.5, let's say we have 35.5 as well, and we have 36. Then it will have 35.67, let's say 48.2, let's say 47, let's say 46.9. Compute the average, we will have 47.37. Let's say we have 51.2, 51.5, 53. Okay, so 51.2, 51.5, and 53 will derive with 51.9. Let's say we have 54.2, 54.3, then we have 53 as well. We will have 53.83, and the negative, of course, 0, 0, and 0. Okay, so these setups are actually the treatments. And these trials will give you the results. Okay? And these trials is actually, or the results derived from the trials are actually based from the experimentations. From the experiments. So that is the main essence of experimentations as a method of data collection in quantitative research. Okay? So, in continuation, we have the method number 3, in which we use surveys and questionnaires. Okay? The surveys and questionnaires. So, from the word itself, surveys and questionnaires, the using of surveys and questionnaires, it uses survey materials. Okay? It uses survey materials. So, to discuss this even further, let us have, okay, another example. Okay, let us have this example, or let us have this discussion. Okay, in surveys or questionnaires, so surveys and questionnaires. Okay, in surveys or questionnaires, it uses the different ranking. Okay, the surveys actually pertain to the ranking or rating, okay, of the different rating or ranking different levels of satisfaction, okay, or agreements. Through the use of different levels of measurement. Okay, so this would be the whole definition of surveys and questionnaires as a method of data collection in quantitative research. So surveys, or the use or the method of surveys and questionnaires, actually pertains to the ranking or rating of different levels of satisfaction or agreements through the use of the different levels of measurement. And what are those different levels of measurement? Okay, you are very familiar with the different levels of measurement already. And that is the nominal scale, the ordinal scale, the interval scale, and the ratio scale. So meaning to say, in surveys and questionnaires, we use these 4 levels of measurement in order to gather different data. Okay? So surveys and questionnaires, since it uses this one, okay, it uses this levels of measurement, meaning to say it is a mixed method of research. Mixed method of research. Okay, meaning to say, surveys and questionnaire uses the triangulation approach as discussed in one of our video presentation. It uses triangulation approach. Okay, meaning to say, when we have triangulation approach, we have different kinds of researches combined together. Okay, so we can have both qualitative and quantitative research when we use the methods of surveys and questionnaires, since it uses names in the nominal scale, the arrangement of these names in ordinal scale, we use continuous data with differences in the interval scale, and we uses different data with 0 as a starting point in the ratio scale. Okay? Also, when using surveys and questionnaires, it not only involves the ranking or rating, it can also provide questions. Okay? Can also provide questions in which these questions involve, okay, yes or no responses. Yes or no responses. Okay? Which will be collected and then counted. So this yes or no responses will be collected and counted. And after the collection, so in this process, we will have the gathering of data. Okay? So after this process of collecting and counting, it will now be subjected to statistics. Okay? So that is the main essence of using surveys and questionnaires as a main method of gathering data in quantitative research. Okay? So now we have the last one. We have the inferential analysis. So actually, inferential analysis is very easy to describe. Okay? So, inferential analysis is actually just the proving of hypothesis. Proving of hypothesis and specific objectives. Okay? Through the use of statistical methods. So, meaning to say, in gathering data, okay, the data will be subjected to the statistical treatments. However, this statistical treatments will provide another data. And that other data will be derived from the method of inferential analysis. Once the data in quantitative research is subjected to the statistical treatments, it will provide another data. And that another data pertain to the method of inferential analysis. Okay? So, the inferential analysis will use the application of the different statistical treatments. And these statistical treatments will actually be seen, or it will be actually discussed in your grade 9 and grade 10 research. Okay, so inferential analysis is the proving of hypothesis and specific objectives. And how do we prove hypothesis and specific objectives? Through the application of statistical treatments. So that is the 4 methods of gathering data in quantitative research. So to recap once more, we have sampling methods in which we derive samples from a given population. Next, experimentation. This is where we form treatments. Surveys and questionnaires. Survey uses survey materials in which the survey materials can, let's say, give ranking or rating of different levels of satisfaction, and it uses the 4 levels of measurement. It also provides questions that answers yes or no, and the answers or the responses will be collected and counted. Now, the 4th method is the use of inferential analysis, in which the gathered data will be subjected under statistical treatments in order to form another data. And this another data will prove hypothesis and specific objectives. And it involves the application of the different statistical methods or treatments. So that ends our discussion for the different methods in gathering data when it comes to quantitative research. So in our next video presentation, we are now ready to discuss to you the purpose of quantitative research.